Hello, I'm Ariel Chipman from the Hebrew University of Jerusalem in Israel. I'm a faculty member at the Department of Ecology, Evolution, and Behavior, where I mostly work on arthropod evolution. I also hold a position as the academic curator of the invertebrate collection and the National Natural History Collections in the Jerusalem branch. And I'm also the Israeli representative for ERGA, the European Reference Genome Atlas. I'm going to talk to you in the next few minutes about biodiversity genomics in Israel, specifically about biodiversity, about genomics, and about the interactions between the two, or as you will see, not that much interactions between the two. So Israel is a small country, but with very high biodiversity. The reason it has very high biodiversity has to do with several factors. One is that it's a continental crossroad. So the Levant, the region that Israel belongs to, the area of southwestern Asia, encompassing Israel, Palestine, Lebanon, parts of Syria and Jordan, since the Miocene at least, has been a crossroads for species moving from Eurasia to Africa and back, many of them staying here along the way. The Levant is also a climatic crossroads, standing as it is in the middle of several climatic regions. So it's connected to Mediterranean climates from the north, to cool steppe and desert climates from the east, to hot desert climates and hot steppe climates from the south and the southeast, leading to a very complex series of climatic zones within a fairly small region. We're talking about a country that's about 500 kilometers north to south and under 100 kilometers at its broadest. This climatic crossroads means that Israel has a mix of fauna and flora coming from different climatic zones. And the most obvious example is the hedgehog. We have three species of hedgehog: a European hedgehog, an Asian hedgehog, and an Arabian hedgehog, whose ranges partially overlaps in some parts of the country. This is true for almost any other group of animals you will look at. We have components from both African, Arabian, Western Asian, and European faunas. The climate in Israel is very complex. So we have Mediterranean climate, steppe climate, desert climates, with a gradient running north to south, and a gradient running east to west from hyper-arid to steppe. So there's a range of different climatic zones mixed with input from fauna from different directions, leads to highly complex and also a lot of local speciation. So because habitats are very patchy and because the climatic gradients are very steep, we can find within very small regions closely related species that have speciated in situ due to patchiness of the habitat. And two examples here are this genus of snails, Sphincterochila, which has 5 or 6 different species with often overlapping regions, and this species of scorpion, Buthus, which also has 6 or 7 different species with partially overlapping ranges. So for its size, Israel has huge biodiversity with a very large number of species concentrated in a very small region. And this is true for almost any group of fauna or flora that we will look at. Israel is also a country with a very strong academic system. There are 8 research universities in Israel, and 3 of them are consistently ranked in the top 100 universities worldwide. We have a very high number of ERC grants per capita. It varies from year to year, but there were a few years when Israel had the largest number of ERC grants relative to its population size. So clearly very good scientists doing very good science. We have many world-class researchers in systems biology, molecular evolution, computational biology, so exactly the type of skill sets that you would be interested in for genomics and comparative genomics. There's also a very strong community studying biodiversity, collection-based research, mostly the Tel Aviv University collections, and a slightly smaller group, but equally active at the Hebrew University of Jerusalem. So given this large biodiversity and this very strong academic system, you would think that Israel is poised to be a powerhouse for biodiversity genomics, but unfortunately that's not the case. Because the two communities, the biodiversity community and the genomics or systems biology communities, don't intersect at all. In fact, there are probably no more than 10 researchers in Israel that really span the range from biodiversity to genomics. And this has not really managed to, has not really turned into a community. So as ERGA representative, I have tried several times, admittedly not with too much effort because this is not my main field of research either, but I've tried to put together biodiversity genomics community get people interested in being involved in IRGA, being involved in biodiversity genomics questions, but this has not been very successful. And usually when you talk to people about that biodiversity genomics, the question they will ask is, can IRGA sequence my favorite organism? The people working on systems biology and comparative genomics are mostly working on bacterial genomes. The people doing molecular evolution are mostly happy working with comparing model system genomes. So there isn't really that much interest. There isn't really that much call for moving into the biodiversity genomics. So what do we need to solve this problem? This is, of course, a big question, and if I knew, then it would have been solved already. But the main thing is that people need incentives to get into comparative genomics. So at the moment, people who are doing biodiversity don't really understand what can genomics do for them, and people working on systems biology aren't really interested in the biodiversity questions, but more in questions of microevolution and the evolution of genome sequence. So we need to find a way to give incentives to study these things. The funding climate in Israel at the moment is not really open to this type of work. I find it difficult to see the main funding agencies in Israel studying projects in biodiversity genomics because these aren't really considered to be leading subjects or high priority subjects. We need to develop greater crosstalk between the communities, convince the systems biology people there's a lot to be done using biodiversity data, and convince the biodiversity people that they can answer the questions they're interested in using genomics. This is partially starting to happen with the development recently over the last 4 or 5 years of a strong community for evolutionary biology, which has representatives from both people interested in biodiversity and people interested in molecular evolution. This is not really happening yet, but hopefully this will get better. But the bottom line is that there is a fantastic untapped resource here. Israel is, for its size and for its academic strength, probably one of the best places to do biodiversity genomics, but there isn't really that much interest. So the question that I face as IRGA representative for Israel, and probably the community faces as well, is how can we tap into this resource and how can we convince people to use it? So I want to thank you for your attention, and I also want to end by acknowledging my student Igor Armiach. The first slides in this presentation were lifted directly from his PhD lecture, and thank you for your interest.